this video, we're going to be looking at a resting membrane potential and how it's generated. A resting membrane potential happens when a cell is at rest. So you notice there's quotation marks around at rest. It means a cell that's not conducting an action potential. Remember, action potential is a movement of two ions. Sodium moves in, potassium moves out to give you an action potential. With the cell at rest, there is a difference in the amount of electrical charge across the membrane, with the inside of the membrane being relatively negative as compared to the outside of the membrane. So looking at the plasma membrane shown here, you have a voltmeter. One of the electrodes is placed on the surface of the membrane outside of the cell. The other electrode is placed into the cell just along the membrane inside the cell, and it registers, in this case, minus 70 millivolts, which means the inside is relatively negative compared to the outside at minus 70 millivolts. That's the difference between the inside and outside. Well, what do we call this potential, this difference in electrical charge between the inside and the outside? It's a cell's resting membrane potential. There's two factors required to generate a resting membrane potential. Number one, you have to have an unequal distribution of ions across the membrane. Shown here is a cell membrane, plasma membrane, and there are ions on both sides, but there's more sodium, for example, and chloride ions outside of the cell. Inside the cell, you have more potassium ions. There's some other ones in here too. We'll mention those later on. But you have an unequal distribution of these ions. The second requirement to generate a resting membrane potential is that it has to deal with the membrane permeability to each type of ion. These ions can't move through the membrane on their own. They can't move through this phospholipid bilayer. They have to have protein channels, and that's what these are here. So the membrane permeability of each ion, they just don't move right across, otherwise there wouldn't be any membrane potential. It has to go through these channels, which are going to dictate the permeability of these ions to go through the membrane. So those are two factors required to generate a resting membrane potential. And what we're going to see later on is that certain cells can send action potentials. They can change their resting membrane potential to send action potentials. A cell that's sitting at minus 70 millivolts, say a neuron, when an action potential happens, that sodium moves into the cell, we're going to see it depolarizes or makes it more positive on the inside. And then potassium is going to move out, positive charge is going to move out, which is going to cause it to repolarize and come back to that minus 70 millivolts. And that's an action potential. Exciting stuff coming up. Let's test your knowledge on resting membrane potential. Pause your video, answer the two questions. Here are the answers. Let's look at each of these requirements for resting membrane potential. Remember, number one is an unequal distribution of ions across the membrane. Sodium and potassium have the greatest effect on the resting membrane potential. You want to star this, highlight this, underline this. There is a greater sodium concentration outside of the cell and a greater potassium concentration inside the cell compared to outside the cell. Well, how do you get more sodium outside and more potassium inside? You have what's called the sodium-potassium pump. Star this also. Almost guaranteed you're going to get a question on this. The sodium-potassium pump is found in the plasma membrane, and it transports 3 sodiums, you can see here, out of the cell for every 2 potassiums that get transported into the cell. Well, in order to do this, you're moving these ions against their concentration gradient. And remember we talked about the cell membrane, that in order to move substances against their concentration gradient, you have to have energy. And that energy comes in the form of ATP. So here's ATP, adenosine triphosphate, and the phosphorylation of that channel provides the energy to move these ions against their concentration gradient, moving sodium out of the cell and potassium into the cell. Well, you might be thinking, well, wait a minute. You said during an action potential, sodium moves in and potassium moves out. Yes, that's true. But you have to maintain this unequal distribution of ions across the membrane for sodium to move in down its concentration gradient and potassium to move out by its concentration gradient. Well, where do we get this unequal distribution from? Sodium-potassium pump. ATP is the energy source required to run the pump, and the energy you burn, the calories you burn to keep your body alive, about 30% of your body's daily energy expenditure is running the sodium-potassium pumps of every cell of your body. Well, there are other ions, chloride ions, calcium ions. These are in higher concentrations outside the cell, the extracellular fluid, but they only have a minor effect on resting membrane potential. You also have molecules such as negatively charged proteins and phosphate ions that you see here. They're inside the cell, and they also have a minor effect on the negative charge that we see inside the cell membrane. But the two ions that have the most effect, sodium and potassium. Let's test your knowledge on resting membrane potential part 2. Pause the video, answer the following 2 questions. Here are the answers. Let's look at the second requirement to establishing a resting membrane potential. You have to have differences in the permeability of the plasma membrane to various ions. These ions cannot cross the membrane without the use of protein channels. Looking at this phospholipid bilayer right here, the plasma membrane, these ions cannot cross this membrane. They need to have channels to cross it. This property of the membrane, to not allow these ions just to move across on their own, allows for the separation of ions and charges that's required for a resting membrane potential to exist. The movement of these ions through various channels, which we have ligand-gated channels, mechanically gated channels, voltage-gated channels, and leak channels. We're going to go into more detail about these channels. For right now, we're just going to focus on the leak channels and their involvement in establishing the resting membrane potential. There are sodium-potassium leak channels, and these allow minor amounts of ions to leak through or diffuse through the membrane down their concentration gradients. And that's what this is shown right here. Here's a sodium-potassium pump. This is a leak channel. They're pretty much open all the time. A little bit of sodium will leak in, a little bit of potassium leaks out, and then that sodium is pumped back out by sodium-potassium pumps, and potassium is pumped back in by, sodium-potassium pumps. The cell membrane, all cell membranes, have more potassium leak channels than sodium leak channels. They're more leaky for potassium than sodium. And what this does is it creates an unequal distribution of sodium-potassium right along the membrane. There are more positive sodium charges along the outside of the membrane than there are positive potassium charges along the inside of the membrane. Even though sodium and potassium both have positive charges, because you have relatively fewer positive potassiums right along the membrane, because the leak channels are allowing potassium to leak out, that gives an overall negative charge to the inside of the membrane. So we say that the membrane's relatively negative on the inside, relative to the outside, because there are less positive charges, less positive potassium than sodium. Remember, you're pumping 3 sodiums out for 2 potassiums in. You also have, like I said earlier, some effect with proteins and phosphates or negative charge. But this unequal distribution of sodium and potassium is the major reason you have a resting membrane potential. And you want to make sure you understand this really important concept. If you need any help on this, please let me know. When we're looking at an action potential, as I said earlier, you have sodium moving in and potassium moves out. And the amount of potassium and sodium, even though it's relatively small, relative to the leak channels, it's a lot. It's like the sodium potassium that moves through this membrane for an action potential is like a faucet here. Well, the sodium leak channels are leaky. A little bit of sodium can move in, but it's like a slow drip compared to this faucet here where it has lots of water coming through. And potassium channels, a little bit more leaky for potassium to leave than a sodium channel, and it looks like this. But it's a very small drip or leak compared to an action potential that involves voltage-gated sodium-potassium channels where a lot more sodium-potassium moves through this membrane. The different concentration of ions across a membrane and the separation of positive and negative charges is a form of potential energy, which is measured in millivolts. And when we talk about potential energy, this is relative to the concentration. For example, if you look at the Shasta Lake here, there's a lot of water behind the dam, and then you have this river coming down on the other side of the dam. The amount of water here is potential energy. You can do work with this water that's behind the dam. In fact, we have these tunnels that lead to turbines, so when the water comes rushing down through these tunnels, it'll turn the turbine and create electricity by the potential energy of the water. The same thing happens with this unequal distribution of ions. Because we have more sodium outside, more potassium inside, when we open up these voltage-gated sodium and potassium channels, sodium rushes down its concentration gradient, moving inside the cell. Potassium then moves through the voltage-gated potassium channels outside, moving down its concentration gradient, and that gives you an action potential. You want to make sure you note that the measurement of voltage is always relative in comparing two regions to each other, such as inside of the cell to the outside of the cell. And that's shown the voltmeter here with an electrode outside and an electrode going inside, and they're comparing these two regions. Inside is relatively negative compared to the outside. Test your knowledge on resting membrane potential part 3. Pause your video and fill in the correct term. Here are the answers. In the resting membrane potential, the inside surface of the cell membrane has an overall negative charge relative to the outside surface of the membrane, which we'd already talked about. So a cell's membrane potential is indicated by a minus sign. For example, this neuron right here has minus 70 millivolts, indicating the inside of the cell's membrane is relatively negative to the outside. And you want to make sure you star this. The resting membrane potential only exists across the membrane, right at the membrane. If you look at the rest of the inside of the cell compared to the outside of the cell, there's just as many positive and negative charges inside as outside, making it neutral for the most of the cell inside and out. It's only along the membrane that we have this difference in charges. And all cells maintain a resting membrane potential. Example, adipocytes shown here, minus 40 millivolts. Thyroid cells, minus 50 millivolts. Neurons, minus 70 millivolts. Skeletal muscle cells, minus 85 millivolts. And only a few cell types, such as neurons, muscle cells, can alter their membrane potential in response to stimulation and send an action potential, which is the movement of sodium in, potassium out, through these voltage-gated sodium-potassium channels, which is referred to as an action potential or neural impulse or muscle impulse, depending on the cell this action potential is moving down. We'll discuss this in a lot more detail soon. Let's test your knowledge. Pause your video, select the term that makes a sentence correct. Here are the answers. That concludes this video, and let me know if you have any questions.